So today we are going to go ahead and look at gene interactions. This is once again Mgomba Ronald, teacher doctor. So we are going to go ahead and look at the different gene interactions, how the different genes interact. Previously we looked at Mendelian inheritance, where we saw the 3:1 phenotypic ratio, F2 phenotypic ratio. We went ahead and looked at the 9:3:3:1 F2 phenotypic ratio of the dihybrid inheritance. We looked at some of the deviations from Mendelian inheritance in our previous meeting that we had. Now today we are going to add onto the gene interactions. So gene interactions are another example of a deviation from Mendelian inheritance. So gene interactions, it's one of the other examples of deviations from the 3:1 phenotypic ratio, or from a 9:3:3:1 phenotypic ratio. So that's what we are going to basically look at, how the genes interact. When you look at Mendel did not know that more than one gene can interact with each other, and each of those genes can influence how they express themselves. So for him, he was looking at discrete units of inheritance. He didn't know that some of the genes interact with one another. So that's what we are going to go ahead and look at. So a single character can be governed by two or more genes. So these non-allelic or intergenic interactions, that's what we are going to be talking about. So it might not necessarily be a gene, a different allelomorphic form of a gene, but sometimes it is a completely different gene, which is on a completely different locus, and interacts and causes some modification of the phenotype. So previously we saw that genes they exist in two allelomorphic forms. At times you might find that we have another gene in a different locus with different allelomorphic forms, but with very many different other combinations, you end up with a different phenotype. That is gene interactions. These interactions can either be, and these independent genes are usually non-homologous. They are located on the same or even different chromosomes. They interact with each other and they express in a single phenotypic trait in the organisms. That's what we are going to be looking at. So we are going to start by looking at the first one, that is known as epistasis. Epistasis involves an intergenic suppression. One gene gets suppressed and as the other is suppressing. So the epistatic gene is the gene that suppresses. So we are going to be looking at two types of genes here. In epistasis, one gene which is located at a different loci or locus masks the expression of another gene on a different locus. Let's take an example, the gene for height, masking the expression of a gene for color. Get that? So that is kind of epistasis. So the one that is usually epistatic gene is the one that suppresses. Then we have another term that we usually use, hypostatic gene. That one is the one that is usually covered up. So a gene or locus which suppresses or masks the action of the gene at another locus was termed as the suppressor, or the epistatic gene, and I've talked about it already. Then the gene which is suppressed by the epistatic gene, that is what they call the hypostatic gene. Take an example. When you look at body color in many of the tropical fishes that we have, is controlled by epistatic interactions between two or more loci, as we are going to see. So we are going to look at the different examples of those genes, of those epistatic genes. Take an example, a set of phenotypes that is controlled by epistatic interactions among 4 different genes. 4 different genes, that means that each of those genes has 2 allelomorphic forms, but they interact in a given way to bring about the different phenotypes that we are going to be seeing in the different organisms. So that is what we are going to basically be looking at. So we have different kinds of epistasis. Yes, we have epistasis. One gene is known as the hypostatic gene. That is the one that is usually masked. Then epistatic gene is the one that is masking the expression of the hypostatic gene. So we have 2 categories. We have those ones which are dominant epistasis and then recessive epistasis. So when you look at dominant epistasis usually occurs when a dominant allele at one locus, that is the epistatic locus, produces a particular phenotype regardless of the genotype of the second locus. So that is dominant epistasis. We are going to make some small crosses to give an example in each of them. So in dominant one, for so long as you have the epistatic gene being dominant, even if it is just one, let's say the presence of capital B is enough to prevent expression of the C's. Let's say we have two genes, gene A and gene B. So this gene B can exist as small b and capital B. Then gene C can exist as small c and capital C. So let's assume that the gene B is the one that is epistatic to gene C. When you look at such an incidence, the gene B, for so long as the presence of a dominant B, capital B, even if it is one, it is enough to suppress the expression of the gene or the other gene, which is C. Whether it is capital C present or small c, it will be suppressed and it will not express itself. So the second type of gene can also express itself in the phenotype only when the epistatic locus is homozygous recessive. This is what I'm telling you. So the other gene, let's say the gene for C, the C, can only express itself when we have small b. When you have capital B, small b in the epistatic locus, you will not see expression of the C gene. So you have for you to see expression of the C gene, you have to lack the capital B, or you lack any B. So that's when you will see expression of that C gene. That is epistasis, and that is the dominant epistasis. So we have some examples of the epistasis, the different types of epistatic interactions of two genes in fish. You can look at them. They are given there. We have the common carp, we have the chinook salmon, we have the goldfish, we have so they are different, and they are different examples. So we can see that this one here, the common carp, is dominant epistasis, duplicate recessive gene interaction. Then here we have recessive epistasis in the Mexican cave characins. And then Sumatran tiger birds, that is duplicate epistasis. So you can see these ratios, these F2 phenotypic ratios are deviating from the normal Mendelian cross. We know the normal Mendelian inheritance cross, the dihybrid F2 is 9 to 3 to 3 to 1. Then here we have 12 to 3 to 1. Here we have 9 to 7. Here we have 12 to 3 to 1. This one we have 9 to 3 to 4. You can see that these are all deviant from the normal dihybrid F2 phenotypic ratio, which is 9 to 3 to 3 to 1. Now, recessive epistasis usually is this kind of epistasis which occurs when the recessive allele of one gene locus, let us say AA, the epistatic locus, suppresses. For so long as you have that small a present, then it will suppress the expression of the other gene. Whether the gene is capital B, the gene, let's say gene B is capital B, it cannot be expressed the moment we have small a somewhere. That is recessive epistasis. The previous one we saw dominant epistasis, where we see for so long as you have the epistatic gene at the gene locus, which is epistatic, for so long as you have a capital B, even if it is 1, it is enough to suppress the expression of the hypostatic gene. So here you just need to have a recessive gene. Double recessive, then it will suppress the expression of the other gene. The other gene, even if it is capital B, it will not express itself. Even if it is capital B, small b, even if it is small b. So that is recessive epistasis. Now, having looked at epistasis, you look at the different examples. You can go ahead and read about more examples of epistatic genes. When you look at even when you look at the inheritance of comb shapes, when you look at the inheritance of comb shapes in the different in foals or in poultry, it also shows some kind of epistasis. So I would like to urge you to go, whenever you have a task ahead of you, you have a question, please read through the stem of the question, then it will guide you and you will be able to identify which type of epistasis it is, and then you can go ahead and make your genetic process to explain. So we can go ahead and look at pleiotropism. Pleiotropism is usually a phenomenon whereby you have one single gene having multiple effects. A single gene having multiple different phenotypic expressions. That is pleiotropism. One gene, it's just one gene, let's say capital B, small b, but it has different ways it can express itself. That is pleiotropism. That's gene interactions we are on. One gene has got its own effect on different parts of the body differently. So let's say in the eyes, it causes for us to have brown eyes. Then when it goes to the skin, it causes you to become black in color. Let's say the gene for melanin. Let's take an example of melanin. When you look at the hair, when melanin is present in your hair, the hair is black. When the gene for melanin is present in your the eye, it will appear brown. When the melanin is present on your skin, it will appear brown. So those are the different expressions of the same gene. That is pleiotropism. You can look at the different examples I've given here, but I've given you examples here. I've talked about melanin, the gene for melanin pigmentation. In your eye, it will appear brown. Then your hair will appear black. The skin will appear brown. So those are the different ways it can express it. So let's take an example of the gene LDBG, which codes for color. This is the gene for color, which looks at the color of the cups. You will realize that it has different pleiotropic effects. Take an example, it will appear blue if you have small b. It will appear gold if you have small g. It will have lowered growth rates. It can even cause lowering of growth rates, for instance, if it is present. Now, the same happens even for albinism when it comes to the cups. That is pleiotropism. Then look at this other one here, which talks about the dominant gene action. Complete dominant gene action occurs when a dominant allele is so strong that it produces its phenotype regardless of the genotype. That is dominant gene interactions. Then we have incomplete dominant gene interaction. We talked about this previously. In incomplete dominance, you see a second type of dominance, but when the dominant allele is incompletely dominant, i.e., take an example. For instance, when you have the snapdragon plant, you have the pink flowers crossed with the white flowers. Then you end up with the red flowers crossed with white flowers, and then you end up with the purple flowers. That is an example of incomplete dominance. The heterozygote is an intermediate, but it is more to the gene that was trying to dominate over the other gene. That is why it is called incomplete dominance. Codominance, we talked about it previously, and we said that codominance, you have two alleles at the locus, and each of them contribute equally to the phenotypic characteristics of the heterozygote. And how it is inherited, it is completely different from incomplete dominance. Because take an example of black and white cows crossed. When they are crossed, you end up with a lone cow, having both white and black fur evenly distributed on the skin of the animal. We can go ahead and look at more. Let's go ahead and look at lethal genes. When you look at lethal genes, usually we call them lethal genes simply because a gene whose phenotypic effect is sufficiently drastic to kill the bearer. When that lethal gene is present, this gene will be sufficient enough to kill the bearer. I will give examples of the epistatic of these lethal genes. Death usually occurs at different. We have different lethal genes that we can talk about. It can happen. It can occur at any time from fertilization to the advanced ages. Take an example of the gene that is in. Let's take. I will talk about examples of when you look at lethal genes, we have dominant lethal genes and recessive lethal genes. But I will talk about one specific example of the lethal genes, and that is found within the mice. When you look at the mice, we have mice which exhibit. We either have the yellow mice or the brown, the yellow mice or the black mice. So when you look at, usually when you get a heterozygotes and you cross them, that is to say heterozygote, and it is usually a brown black mouse. When you cross them, you will end up with a ratio of black and yellow. You will end up with a 2-to-1 phenotypic ratio. I asked yourself a question, but I'm ending up with a 2-to-1 phenotypic ratio. This one is giving me a total ratio of 3, yet I'm expecting a total ratio of 4. A 2-to-1 phenotypic ratio is showing us that the one of them, which was 1, the one that would be 1 to making to be 3-to-1, So the one died during embryonic development. That is to say, when you look at those genes, there are two. We have the capital Y and the small y. If you have a heterozygote, imagine if you have a heterozygote, you'll have capital T, capital Y, small y, crossed. And when you have capital Y, small y, and you make your cross, you'll have with a genotype with capital Y, capital Y, capital Y, small y, and then small y, small y. Now, capital Y, those ones, because it is dominant litho gene, the capital Y kills the beholder. And those rats they are never born. And then we end up with the capital Y, small y, that is the dominant one. That is the heterozygote. Capital Y, small y is heterozygote. And then small y, they are the yellow or albino rats. So you end up having all the black rats that are existing are maybe, let's say, they are all heterozygotes. Why? Because the litho gene, presence of the litho gene in its homozygous state, it kills the beholder. That is an example in albino rats. Take an example in tilapia. We have another example of litho genes in tilapia. You can see here that if you have capital S, that one kills the beholder. If you have capital S, small s, it will survive. That is normal. Then small s, or what they are calling plus. This one here is small s. This one will be normal and it will survive. This one small s, capital S, this one will be normal. This one will be abnormal fin, but it will not survive. It will survive still. But when you look at SS, those ones will die. So this SS, even in the mice, the capital S in the mice, sorry, capital S in the mice, all what we call capital Y in the mice, these ones for them, they will die. And you end up with the capital S, small s. So that's why you end up with a phenotypic F2 phenotypic ratio of 2:1. So that means that you have a deviation from Mendelian inheritance. So that is dominant recessive or dominant litho genes. Then we can go ahead and conclude with the recessive litho genes. Now, recessive litho genes, for them, it can only express itself when we have when both of them are recessive. When the gene that is present is recessive, then it can cause death. Let's say in the guppies, when you look at the guppies, you realize that when the Y chromosome are combined with a male, the homozygotes with respect to the genes, they will appear homozygotes. So just remember that in litho genes we have two different categories. That is to say, recessive litho genes and the dominant litho genes. The dominant litho genes, presence of the capital T, or capital Y, or capital S kills the beholder. In the recessive, the presence of the recessive gene will kill the beholder. So that is basically what we talk about in litho genes. Just to remind all of us here, we talked about the different sex-linked litho genes. We talked about sex linkage, where we said a gene which has nothing to do with sex determination is being carried on the sex chromosome and is inherited along with the sex genes. We call that sex linkage. And we saw the different examples of sex linkage. Do you still remember the different examples of sex linkage? Please mention them.
We talked about sickle cell.
Sickle cell is inherited in a codominance fashion or incomplete dominance fashion. In fact, most textbooks, they will say it is codominance. So sickle cell is not sex linked. But we have blue-green color blindness. We have hemophilia. All those ones are examples of color blindness. Okay? Hemophilia, blue-green color blindness. All those ones are examples of sex linkage. So we can also have litho genes which are sex linked, whereby if they are present, they will kill the individual having them. We talked about multiple alleles, and we said in multiple alleles, it is a type of interaction whereby it is not known to Mendel, and we say that one gene being expressed in more than 2 allelomorphic forms. But we said at any given time at the locus, you'll only have 2 different forms. You will never have more than 2 different forms at any given time. And we gave an example of multiple alleles, that like inheritance of fur color in the rabbits, or inheritance of blood groups. They show multiple alleles. So that is all about, when you look at that, is all about multiple alleles. Sometimes we have what they call complementary genes. Now, complementary genes are like genes when they are present, they complement, they allow the other genes to express themselves very well. That is what we call complementary genes. A complementary gene can, when it is present, it will allow other genes to express itself, and you end up with a 3 to 9 phenotypic ratio, where you have you expected a 9 to 3 to 3 to 1, you end up with a 9 to 7 phenotypic ratio. Why? Because both dominant alleles, when present, capital A, capital A, capital B, capital B, capital A, and so on, and complement each other. They complement each other. So that example is what we usually call complementary interaction, gene interaction. It is somehow related to what we call epistasis. So that in epistasis, the presence of one gene at the different locus suppresses the expression of the other gene, known as the hypostatic gene. So this one here, it just amplifies the expression of the other. That is complementary genes. So thank you so much. I think that is all about gene interactions.